Good afternoon. Welcome to Elimu Live KUTV. It's Biology Form 1 again, and we are doing a continuation of nutrition in plants and animals, and digestion in the small intestine is our objective for today. So welcome and feel at home. And if you have any questions, you can post them. There are the numbers on your screen. Also, feel free to share with all your friends, and everything will be fine. So last time we had this particular class, we discussed digestion in animals, and we discussed digestion in the mouth, and we also discussed digestion in the small intestine. With me is a picture that shows the digestive system, and we're going to recap what we learned, then we can continue what we're going to learn today. So this is a picture that is showing the digestive system, and the digestive system is actually a tube that runs from the mouth down to the anus. So from here we have the mouth, then we have a tube that connects the mouth to the stomach, and that tube is called the oesophagus. Then from there we have the part that is here, which is like an accessory organ to the digestive system. This is called the pancreas. It is shaped like a leaf. Then we also have the liver, which is connected to the first part of the small intestines. The first part of the small intestine is called the duodenum. So this portion between this and this is the duodenum. Then we have the lower part of the small intestine called the ilium. Then on this side, we have the large intestines. From there, we have the rectum and the anus. So our objective for today will be looking at digestion in the small intestines, and we're going to look at how the mechanical and chemical digestion takes place. We're also going to look at the enzymes and how they help in the breakdown of food substances. So before the food gets into this part called the small intestine, it first gets to the stomach. Now from the stomach, there is chemical and mechanical digestion. So this chemical and mechanical digestion, it breaks down food to a paste-like substance that is called chyme. So chyme is the one that is going to enter into the small intestines. The small intestines are divided into two parts. The first part is called the duodenum, and the second part is called the ilium. So these parts, the duodenum is usually on the upper side, and the duodenum receives secretions from the liver and the pancreas, while the ilium is on the lower side. Now, when you're writing the word ilium, make sure that you check your spelling. There's usually a likelihood of students writing ilium with a double L as a done, and this costs you a mark because that's wrong spelling. So ensure that anytime you have the word ilium, you just write it with one L. Now, sometimes in the examination, you might not be able to remember how to write the word ilium. So if you just write small intestines, you'll also get a mark for the same. Now we're going to look at digestion in the duodenum. So as I said earlier, the duodenum is the first part of the small intestine. So the duodenum is right here, and it receives secretions from the liver and the pancreas. So this portion is what we're calling the duodenum. So these particular organs are the ones that secrete chemicals that assist in the breakdown of the chyme. The first organ, we have the liver. So the liver is the largest internal organ of our bodies, and this liver secretes a substance, but before it secretes a substance, there is usually a hormone produced. Now, a hormone is a substance that initiates a process. I repeat, a hormone is a substance that initiates a process. So presence of food in the duodenum will cause the secretion of a hormone called secretin. Now, secretin will now cause the liver to produce bile juice. It stimulates production of bile juice by the liver. So this particular bile juice has components that will enable digestion to take place. Now, when you are defining, or when we define the term digestion, we say that digestion is the process that food substances which are complex are chemically and mechanically broken down into soluble substances. I repeat, digestion is the process that complex food substances are chemically and mechanically broken down. So this bile juice helps in those two processes. It helps in chemical breakdown. It also assists in mechanical breakdown. So I hope we are moving on together, and it is always wise to take some notes so that maybe you refer when you forget some of these concepts. So we are going to look at the bile juice. So the bile juice is produced by the liver. It's produced by the liver, and the bile juice has some salts. It has some salts inside it. So these salts are two, and the first salt is called sodium glycocholate. The second salt is called sodium taurocholate. So these two are what compose the bile salts. So these salts, their main function is emulsification. These salts have a main function of emulsification. So what is emulsification? Emulsification is a simple process that involves breakdown of large fat droplets into tiny fatty droplets. These are large fat droplets into tiny fat droplets. So what is the importance of emulsification? Emulsification is vital because it helps to break down the fats into smaller substances, and this increases surface area for digestion of fats. I repeat, it helps in breakdown of large fat droplets. It helps in breakdown of large fats to tiny droplets to increase surface area for breakdown of fats. Now, this process that we're calling emulsification is a mechanical process. Now, mechanical means it does not involve any chemicals. And it is a mechanical process because these particular fats are not going to be broken down into their components. Now, in the previous subtopics, we have learned about the food components or the chemicals of life. So we have carbohydrates, we have proteins, we have vitamins, and we have fats. The building blocks for fats are, number 1, glycerol, number 2, fatty acids. So this particular process has not broken down the fats into glycerol and fatty acids. It has broken down the fats into droplets that are tiny. So just get that difference, that this is not a chemical process, but it is a mechanical process. Now you can find sometimes in the exam you brought a diagram. For example, you brought these two, then there is an arrow, then there are this portion of tiny fat droplets, and then you asked identify the process. So the process is emulsification. And then sometimes in the exam you can be asked what is emulsification. So maybe you can write that down as you write the answers. What is emulsification, or what is meant by the term emulsification? So what is emulsification? What is emulsification is the process that large fat molecules are broken down into tiny fatty droplets. So the keywords here, we have large fat molecules that must be present. Also, what are they broken down into? Tiny fatty droplets. So I hope we're moving on together. So that is one and the main function of bile salts. And we've said there are two, that is sodium glycocholate and sodium taurocholate. So from this you can be asked, what is the significance or what is the importance of emulsification? What is the significance or importance of emulsification? What is the significance? So the word significance means what's the main thing that this particular process helps in. So it is to increase, it helps to increase surface area for breakdown of fats. Increases the surface area for breakdown of fats. So from there, the bile juice also has another function. We say that when food leaves the stomach, it leaves the stomach in form of a paste, and that paste is called chyme. Now this paste has some acidic properties. Why does the paste have acidic properties? It has acidic properties because the paste is leaving the stomach. And in the stomach, we have hydrochloric acid. Now, this bile juice helps to neutralize hydrochloric acid in chyme. So that's the other function of bile juice. Apart from emulsification, it neutralizes acidic chyme. The word neutralize means it reduces the effect of this particular acid called HCl. Neutralizes, bile neutralizes acidic chyme. So that is it. So if you ask for two functions of bile juice, neutralizes acidic chyme and also it helps in emulsification. So I hope up to that point we are understanding. And from this picture over here, this is the liver and it is the one that helps in production of bile juice. Now let us move on to the next organ that produces secretions that assist in the digestive process. So the next organ is right here, and that organ is shaped like a leaf. It is highlighted with a yellow shade. So this is the pancreas. So the pancreas helps in digestion by removing a secretion. So the pancreas removes a secretion called pancreatic juice. Now pancreatic from the word pancreas. So pancreatic juice has some components. It has some components. Pancreatic juice is made up of 4 main components. So components of pancreatic juice. Components of pancreatic juice. So the first component of pancreatic juice is called pancreatic lipase. Pancreatic lipase. Pancreatic lipase. The second component of pancreatic juice is called pancreatic amylase. Pancreatic We also have the third one that is called trypsin. And finally, we have sodium hydrogen carbonate. Sodium hydrogen carbonate. So these are the components that make up the pancreatic juice. Pancreatic lipase, pancreatic amylase, trypsin, sodium hydrogen carbonate. Now, from knowing the components, you're now supposed to know the functions. What are the functions? Now, these components now assist in the second part of digestion, that is chemical digestion. How do they assist in chemical digestion? We're going to start with the first component, which is pancreatic lipase. Now, from the word lipase, we take the first letters, lip. Now, the first letters have been derived from the word lipids. Lipids. Now, lipids are fats and oils in biology. So, how does this enzyme assist? Now, pancreatic lipase is an enzyme. What is an enzyme? An enzyme is a substance that speeds up a reaction or a process. An enzyme is a substance that speeds up a reaction or a process. So pancreatic lipase speeds up the breakdown of lipids, and these lipids are now split into their constituents, that is fatty acids and glycerol. So they are broken down into fatty acids and glycerol. So if you're writing, we can write this: pancreatic lipase speeds up the breakdown of lipids to fatty acids and glycerol. So fatty acids and glycerol are the components of lipids. Now we move on to the next component, pancreatic amylase. Now pancreatic amylase, it's usually an enzyme that helps to complete a process that was started in the mouth. So I'm going to take you back to the mouth. Digestion begins in the mouth. And presence of food in the mouth stimulates the salivary glands to produce saliva. Saliva has three components: water, mucus, and salivary amylase. What is the function of salivary amylase? It speeds up the breakdown of some starch to maltose. Now, starch is a complex carbohydrate, so some of it is broken down to a lesser complex carbohydrate called maltase. Now, this some starch, not this some starch. So where does the other starch go? The other starch is taken down the oesophagus into the stomach, then from the stomach it is taken to the duodenum. So this remaining starch, the breakdown is made possible by this enzyme we're calling pancreatic amylase. So when you're defining the function of pancreatic amylase, you should include the word remaining starch, because the work had already started in the mouth. So pancreatic amylase speeds up breakdown of the remaining starch to maltose. So I hope we're moving on together. In case you have a question, we have numbers on your screen. You can ask your question. In case you have a comment, we'll appreciate your comment. Also, feel free to share this content with all your friends who take biology. So we have another component of pancreatic juice, that is trypsin. Now, before we get to the function of trypsin, I'd like to emphasize that trypsin is also an enzyme. And trypsin is not produced as trypsin. It is produced or secreted in an inactive form. It is secreted in an inactive form. So the inactive form that trypsin is secreted is called trypsinogen. Trypsinogen. Now, why is it secreted in an inactive form? Now, trypsin is an enzyme. Then we have this part called duodenum, which has walls. The walls have some components made up of protein. Now, in case it's released as trypsin, it will start speeding up the digestion of those proteins. So for it not to digest or corrode the wall of the duodenum, it has to be secreted in its inactive form called trypsinogen, right? So trypsinogen is now, when trypsin is in action, trypsinogen ceases to function. Like, you cannot use trypsinogen, you can only use trypsin, right? So the work of trypsin, it speeds up the breakdown of proteins to peptides. Now, proteins are complex. They have to be broken down to simpler forms. So the peptides are the simpler forms. So speeds up, again, don't forget the word speed up. Speeds up breakdown of proteins to peptides. Speeds up breakdown of protein to peptides. Now, you'll find that in some books, they don't write the word peptides. You'll find that they use the word peptones. So the word peptones and peptides means the same thing. So the last component of the pancreatic juice is sodium hydrogen carbonate. So sodium hydrogen carbonate is just a substance. It is not an enzyme. I repeat, it is just a substance, but not an enzyme. So sodium hydrogen carbonate has one main function. It assists the bile juice in neutralizing the acidic chain. It assists the bile juice in neutralizing So it neutralizes acidic chyme. Neutralizes acidic chyme, right? So this component called sodium hydrogen carbonate also has another function. Now, sodium hydrogen carbonate is alkaline in nature. So it provides a suitable environment for the working of pancreatic enzymes. Now, enzymes are specific in the environment they work in, or they are specific in the medium they work in. Some enzymes function best in acidic pH. Some enzymes function best in neutral pH, while other enzymes function best in alkaline pH. So the pancreatic enzymes function best in alkaline pH. So sodium hydrogen carbonate provides an alkaline environment for the working of pancreatic enzymes. So up to that point, I hope we're moving on well, and I also hope you're understanding these concepts. So from this part, duodenum, we move to the last portion of the small intestine, which is the ilium, and this is where digestion process ends. So we're going to look at what takes place in the ilium, and we're also going to look at what secretions are produced there and the functions of these secretions. So ilium. The ilium is the lower part, or the last part, or the last portion of the small intestines. Ilium, last portion of the small intestines. Now, presence of food in the ilium will cause the walls of the ilium to produce or to secrete a juice. I repeat, presence of food in the ilium will cause the walls of the ilium to secrete a juice. So this juice that is secreted by the walls of the ilium is called intestinal juice. Intestinal. It is secreted by the walls of the ilium. Now, this intestinal juice also has a scientific name, and the scientific name for intestinal juice is Succus entericus. Succus entericus. Remember, when we are writing the scientific names, always observe the principles of binomial nomenclature. That is, the first word, which is genus, must always start with a capital letter, while the second one, which is the species name, should always begin with a small letter. Also, do not forget to underline them separately. So this intestinal juice has components, and intestinal juice is purely made up of enzymes. So we are going to outline these enzymes, and we are going to see how they help in the process of digestion. So the first enzyme is maltase. The second enzyme is sucrase. The third enzyme is lipase. The fourth enzyme is peptidase. And the last enzyme is lactase. So these are the components of the intestinal juice: maltase, sucrase, lipase, peptidase, and lactase. So in the examination again, you can just find a simple question asking you, state 3 components of intestinal juice. So you can state any. While you're writing them, there's no specific order of stating them. So maybe in that particular exam, you are remembering peptidase. Is not wrong if you start in any order. So what are the functions of these enzymes? Again, I will remind you. An enzyme is a substance that speeds up the rate of a reaction. So anytime you're writing, revising, answering any question about an enzyme, make sure you include the word speed up. So we're going to start with maltase. Now, from this word maltase, we get a chemical compound called maltose. Now, maltose is a carbohydrate. So this maltase is an enzyme. And when we're learning about characteristics or features of enzymes, we say that enzymes are specific to substrates. Enzymes are specific to substrates. What does that mean? Enzymes are specific to what they act on. For example, maltase from the word maltose will only speed up the breakdown of maltose. Sucrase from the word sucrose will only speed up the breakdown of sucrose. Lipase from the word lipids will only speed up the breakdown of lipids. Peptidase from the word peptides will only speed up the breakdown of peptides. And lastly, lactase from the word lactose will only speed up the breakdown of lactose. So I hope you understand that point of being specific. So we're going to start from here. What is the function of maltase? Maltase usually speeds up breakdown of maltose into 2 components. The 2 components of maltose are glucose and glucose, or just glucose alone. Are we together? Glucose plus glucose is maltose, right? So it just splits maltose into glucose. So speeds up breakdown of maltose to glucose. Speeds up breakdown of maltose to glucose. Then we have sucrase. Now from the word sucrose, it speeds up the breakdown of sucrose to 2 things, glucose and fructose. I repeat, it speeds up the breakdown of sucrose to glucose and fructose. Speeds up breakdown of sucrose to glucose and fructose. Glucose and fructose. Some people prefer to call it fructose. Whichever the case, just make sure in the exam you can write it using the correct spelling. So we have the 3rd component, lipase, from the word lipids. So lipase, it speeds up the breakdown of lipids to 2 things. Number 1, glycerol. Number 2, fatty acids. Now, a lipid is made up of those 2 components. It's made up of the glycerol, which is usually shaped like this, also known as the triglyceride. Then it is usually joined to 3 fatty acids. So this is a lipid. So it is now broken down. So this bond is broken down such that we are now going to have the fatty acids alone and the glycerol alone. So I hope you're getting me. So it speeds up breakdown of lipids 2 fatty acids and glycerol, just as you say it, glycerol. We have peptidase. Peptidase is also an enzyme. Now, peptidase, it speeds up the breakdown of peptides to amino acids. It speeds up breakdown of peptides to amino acids. So peptides are units of proteins, but the building blocks for proteins are amino acids. So amino acids are the most simple form of proteins that can be absorbed by the body. So this breakdown is usually made possible by enzyme peptidase. Lastly, we have enzyme lactase. So from the word lactose, it speeds up the breakdown of lactose into two components. Number 1, glucose. Number 2, galactose. I repeat, it speeds up the breakdown of lactose into two components. Number 1, glucose. Number 2, galactose. Now, sometimes students get confused between sucrase and lactase. And the confusion comes in with the function. Now, if I write the word galactose, galactose, so from this lac and lac, you can be able to relate and differentiate what does what. So, lactase speeds up breakdown of lactose to glucose and galactose. So these are the main components of the intestinal juice, whose scientific names is Succus entericus. Don't forget that anytime we talk about an enzyme, we usually answer it by speeds up, right? So up to that point, I hope you're getting what we are discussing today. In case you have questions, there are numbers on your screen. Also, feel free to share this content. I know it will be of assistance. Now, when these complex substances have reached the ilium, that is where digestion stops. And the substance that is formed after digestion in the ilium has taken place is called chyle. The substance that is formed is called chyle. So chyle is composed of the simple substances we talk about from the term digestion. So digestion is the chemical and mechanical breakdown of complex food to simple soluble substances. So this chyle is made up of the simple soluble substances, for example, glucose, for example, amino acids, and also fatty acids and glycerol. Absorption will now take place. Absorption is the process that these soluble substances get into the bloodstream. Now, for them to get into the bloodstream, there are some adaptations that the small intestines possess. Now, what is an adaptation? What is an adaptation? An adaptation is a special feature. I repeat, an adaptation is a special feature. It is a structure that makes an organ unique. So we're going to look at adaptations of the small intestines to their function. Now, the small intestines, which are made up of two parts, duodenum and ilium, have two main functions. Number one, digestion, and we have seen the enzymes that assist in digestion, and because they are enzymes and they are chemicals, they assist in chemical digestion. We also have absorption. So these soluble substances are taken in. So the first adaptation of the small intestines, first adaptation, is that it is long. So with me is a cable, and this cable is long. So the small intestines are very long. In fact, in some adults, if they're stretched, they can run up to 6 meters. So being long, it has increased surface area for digestion and also absorption. Now, if this cord is short, the surface area is less. But if it's long, the surface area is more. So the reason why they are long, to increase surface area of digestion and absorption of digested food or soluble food substances. So anytime you give an adaptation, you must give the function. How does it assist? If you get an exam asking you, state two adaptation of the small intestines to their function. Now, two adaptations means that you're going to write the adaptation and also explain the function. Now, in case you have an exam and you just write, it is long, you leave it that way. You don't score a mark because we have many long things in biology. So why is it long? How is this assisting in the functions? So always be sure to give how it assists in the function. The second adaptation of the small intestines is that the small intestines are highly coiled. Now, we've talked about 6 meters. So imagine if our intestines were not coiled, like we'd be tall, think we'd reach the sky. But they are highly coiled. So if these are your intestines, they are usually coiled. So it coils there, it coils there, it coils there, coils there, coils there. So highly coiled means that the coiling is maximized. So they are highly coiled for 2 reasons. Number one, for them to be able to fit into your abdominal cavity. And number 2, to slow down movement of food in order to allow more time for absorption and digestion. Now, if these particular intestines were just not coiled, if I stretch this and maybe I had space in between and I put some water inside, it would flow very fast. But if I coil it, the water would flow slowly because it is coiled. So the more the coils, the slower the movement of the food substances. So this will give time for the succus entericus or intestinal juice to take its function. It will also give more time for absorption of the soluble products. Hope we are clear on that. So when you explain this point, you must capture to slow down movement, slow down movement of food, giving more time for digestion and absorption. And don't get tired of explaining. You always, when you sit for an exam, always assume the examiner does not know this concept. So do your best to put your words in the simplest form possible. So those are a few of the adaptation of the small intestines to their function. There are more adaptations. For example, they have an inner lining that is called the epithelium. Now this inner lining has very thin layer of cells. So this allows faster diffusion of end products of digestion. Now, I want to leave you with this one concept. When you're talking about absorption, you don't talk about food. Avoid using the word food alone. Don't talk about food. This is wrong. You either talk about digested food or soluble end products of digestion. You talk about digested food or soluble end products of digestion. So I hope we're getting each other, and I hope you're also underlining the main points. So up to that point, we have achieved our objective of looking at how digestion occurs in the small intestine and the things that you should consider. What part is what, because you can be brought that diagram in the exam and asked to label the parts. So know the parts. Number 2, know what is secreted by what organ. Also, know the function of every enzyme at every particular stage. So I will just leave you with 2 questions for assignment. Number 1, name 4 components of the intestinal juice, 4 marks, and state the enzyme that stimulates secretion of bile juice. 1 mark. So do as many questions as possible, then you'll get to understand this concept. I have been your host, Helen MUTHONI NGARI. Thank you for watching us. Have a blessed one. Thank you.